Good morning, everyone. I would like to thank the organizing committee for giving me the opportunity to present my work here. This is a work done in collaboration with the research group in Germany, Munich. Dr. Habig did this ex vivo perfusion of transgenic pig hearts. And my talk will be about the evaluation of the innate immune activation after the xenoperfusion of GAL knockout CD46 HLA transgenic hearts. So as a background, cardiac xenotransplantation may be a solution for the shortage of human donor hearts. And genetic modification, of course, are required in order to inhibit activation of the complement system through the known anti-GAL antibodies. So GAL knockout and human CD46, at least for heart transplantation, are necessary. However, early xenogenic reactions are mediated by various leukocyte subsets, and one of those are natural killer cells. So there are several lines of evidence which suggest that NK cells are involved in xenograft rejection. And as you can see here, human NK cells are able to adhere to and lyse porcine endothelial cells in vitro. Also, in ex vivo perfusion, it has been shown that they infiltrate pig organs, for example, in a pig limb perfusion, in a heart perfusion, and also in lung perfusion. And these are really recent studies from 2017. Also, primate NK cells are able to infiltrate pig to non-human primate xenografts, and also it has been shown to have a role in small animal models, also in xenotransplantation. So, human NK cells are able to kill other cells through two different mechanisms. One is the antibody-dependent cytotoxicity, and the other one is the direct cytotoxicity by recognition of antigen on the cell surface. So this is what's depicted here is a typical situation when wild-type organs are perfused with human blood, for example. You have that it's a matter of balance between inhibitory and activator receptors. So if the activator signals are more and more powerful, you get activation of NK cells. So they start to release granzyme and perforins, and then you have killing of the cells. If you have engagement of inhibitory receptors, you have inhibition of the NK cells action. So there is a lack of inhibition by porcine MHC class 1, which is called also spine leukocyte antigen. And the activation, unless we have the activation of NKG2D, which is an activating receptor, and NKP44 on NK cells. So we have the killing of porcine cells. Protection from NK cytotoxicity might be obtained thanks to the expression of human MHC class 1, in our case HLA-E, on porcine endothelial cells in transgenic pigs. So the aim of this study was to test the potential of the combined overexpression of human CD46 and HLA-E to prevent both complement and NK cell-mediated xenograft rejection in a pig-to-human ex vivo cardiac xenoperfusion model. So this is the method which Jan performed. So we used 6 genetically modified pigs, and 6 wild-type pig hearts were used as a control. So hearts were perfused for a total of 8 hours with heparinized human blood, 3 hours in a working mode, and then 5 hours in Langendorf perfusion. After the experiment, biopsies from this myocardial tissue were obtained, and then immunofluorescence on frozen sections were performed. We evaluated transgene expression, complement deposition on the tissue, immunoglobulin binding, IgG and IgM, coagulation factors, so tissue factor expression on the tissue, fibrin releases marker like TPA and PI1, and NK cells infiltration through the NKP46 marker. So as you can see here from the pictures and also from the quantification, human CD46 is overexpressed in the transgenic hearts and also HLA-E, while they are absent in the wild-type hearts. The alpha-gal is expressed only in wild-type hearts. And this is the quantification of the fluorescence intensity. So we looked at complement disposition, so in particular C3bC and also C4bC on the tissue, and immunoglobulin binding, IgM and IgG. And we saw that there is a significant decrease of complement deposition thanks to the overexpression of human CD46. And then also, this is the case also for the immunoglobulin binding. We looked also at the fibrinolysis and the coagulation markers. So Pi1, TPA, tissue factor. We didn't observe a difference in tissue factor expression, TPA, but we saw a slight decrease in the Pi1 expression. And lastly, we looked at NK cells infiltration in the tissue. Through the staining of NKp46, we were able to observe and quantify the presence of NK cells in the myocardial tissue. And after quantification, we saw that in the genetically modified hearts, we observed less NK cell presence. So in conclusion, overexpression of human CD46 led to a reduced myocardial complement deposition by blocking the formation of C3b and C4b. Immunoglobulin binding was also affected and reduced in the genetically modified hearts. Expression of HLA-E resulted in reduced NK cell migration into the xenogenic tissue. This model is able to assess NK cell migration after 8 hours of perfusion with human blood and might be a helpful tool to further assess the role of other transgenes in preventing innate immune cell-mediated rejection. With this, I would like to acknowledge my supervisor, Rob Driven, my co-advisor, Jörg Sebak, and also the great work which has been done from the group of Professor Riccardo and also Jan Abicht, which he performed the xenoperfusion experiments, and also the other members of my lab. Thanks a lot for your attention.
Thank you very much for this interesting talk. I do have 2 questions. Did you also use Gartenocracy 46 alone without the additional expression of HLA-E to assess separately what the HLA-E does by itself? And the second question would be, did you see any difference in performance of the organ, cardiac output or contractility?
So, regarding the first question, we didn't have organs from human CD46 gall knockout pigs. But we would like to try in vitro, because we have cells from these pigs. So our aim now is to test in vitro this genetic modification and see the effect of the HLA, at least in vitro, and then maybe in the future try xenoheart perfusion with this kind of transgenic modification. So regarding the second question, could you please?
The difference in performance of the heart?
Yes, there were differences. I'm not an expert about this. Maybe Jan is not here. He already showed some data in the previous congresses about the heart from the genetically modified pigs. They were more healthy, let's say, at the end of the perfusion. We can see also from the histology it was more preserved. The morphology was preserved.
Any other questions? Yes, Peter?
You showed there was a reduction in NK cells with the transgenic.
Yes.
How many are there to begin with? Are there many NK cells in the control, or is it just the occasional cell that you find?
Okay, we tried also to do this kind of staining in other biopsies from a myocardial infarction that we have. And we tried in an area not at risk, so healthy tissue, and we didn't find any staining of NK cells. We tried also in necrotic ischemic tissue after myocardial infarction, and we were able to find also porcine NK cells in the tissue, because this antibody is cross-reacting with porcine antigen. So I think we took a lot of different spots in the biopsy that we had, and we were able to see some spots were more infiltrated with the NK cells compared to other.
And are they associated with other cells, or are they just alone?
This we didn't try a double staining, but this will be interesting. Yeah. Thank you.
Yes, David.
Did you show the expression rate of these two molecules compared with human ones? I hope to the expression rate. How
Okay. Yeah, no, we don't. I didn't show controls, but we did staining of, for example, human CD46 in human cells, and we saw that in this transgenic peak, the expression is really high, so it's overexpressed.
I also, of course, not interested in the expression. Used to be Java, we argue how to express HLE. HLE is difficult to express. Then I hope to know the expression rate of HLE compared with the human.
This, I cannot answer this question. I know that it's difficult to have expression of HLA-E. That's why also these peaks are transgenic, and these also include the beta-2 microglobulin to have a better expression of the HLA-E.
So I think concerning this question, those peaks were generated, I think, by IXA, if I'm not mistaken. And Riccardo has also used the same genetics, and it was tested, and the HLA-E expression was actually very high at those.
Mm-hmm.
Thank you very much.
Thank you.